Welcome to the Deep Dive. Today, we're tackling something absolutely fundamental, how life on Earth actually gets its power.
Right. Where does the energy come from?
Exactly. We've talked before about breaking down carbs for energy, but today we're closing the loop. How does that energy get into the system?
That's a great question.
Our mission here is to really unpack photosynthesis using a key chapter from the Raven biology of plants as our guide. Think of it as your shortcut to getting this core process.
And it is core. We're talking about the engine that fuels almost everything. Billions of tons of sugar made every year.
All inside these tiny chloroplasts. It's basically how life fights off, well, chaos, entropy.
And for you listening, we want this deep dive to connect the dots, clear up maybe some old misconceptions you might have.
Hopefully give you a few aha moments.
Exactly. We'll trace how we figured this all out, from, ancient ideas right up to modern science. The chapter lays it out nicely. History, light, pigments, then the nitty-gritty reactions.
So it's a journey of discovery. Let's get started.
A historical perspective.
Okay, first stop, history. And it starts with a bit of a mistake, really. The ancient Greeks, like Aristotle.
They saw animals eat, right?
Right. So they figured plants must do the same, get all their food, their substance, straight from the soil. Seems logical, but...
Totally wrong, as it turns out.
Enter Jan Baptista van Helmont around the 1600s. He decided to actually test this. Planted a willow tree in a pot.
Carefully weighed everything first.
Yep. Weighed the tree, weighed the soil. Then, for 5 years, he only added water. Nothing else.
And the results were pretty dramatic.
Weren't they just? The tree gained over 74 kilograms. Huge increase. The soil. It lost barely 50-something grams. Practically nothing.
Van Helmont thought, aha, it must be the water. He concluded the plant's mass came from water.
Which is partly true. Water is involved.
But it's not the whole story. He completely missed the role of the air, specifically carbon dioxide. Couldn't measure it back then.
Okay, fast forward about 150 years. Joseph Priestley, an English clergyman, actually.
And he did these fascinating experiments with candles and mice in sealed jars.
Right. He found if you burn a candle in a sealed jar, the air gets injured, as he put it. The candle goes out. A mouse can't live in it.
But then,
if you put a sprig of mint in that same jar for a few days, voila, you could light another candle, a mouse could live again. The plant had somehow restored the air.
Which in modern terms means plants take up CO2 and release oxygen?
Exactly. He stumbled onto the basic gas exchange that keeps us all alive.
Then came Jan Ingenhousz, a Dutch doctor.
He confirmed Priestley's findings, but added crucial details. This restoration only happened in sunlight.
And only with the green parts of the plant. Light and green stuff were key.
He, like others, guessed that maybe the plant split the CO2 to get the oxygen.
That seemed logical for a while, but then C.B. van Niel came along and shook things up.
How so?
Well, he was studying these purple sulfur bacteria. They do photosynthesis, but they use hydrogen sulfide, H2S, instead of water.
And they don't make oxygen?
Nope. They release sulfur, pure sulfur. So van Niel looked at this and proposed something radical. Maybe in all photosynthesis, the oxygen comes from splitting the water molecule, not the CO2.
That's a big leap. Water giving us the oxygen we breathe.
It was. And the evidence started rolling in. Robin Hill, in the 30s, showed chloroplasts could make oxygen with light, even without any CO2 present.
The Hill reaction.
That's right. But the clincher came in 1941. Ruben and Kamen used heavy oxygen, an isotope called 18O.
How did that help?
They gave plants water labeled with this 18O, and guess what? The oxygen gas the plants released also contained 18O.
So the oxygen definitely came from the H2O.
Exactly. Case closed, water is the source of O2.
So the balanced equation we use now reflects that. 3CO2 plus 6H2O
Using light energy?
gives you a basic sugar, C3H6O3 plus 3O2, and interestingly, 3H2O.
Right. Water shows up on both sides.
Yeah.
Some is consumed, split apart, and some new water is actually formed during the process of making the sugar.
And we should also mention F.F. Blackman back in 1905.
Good point. He showed photosynthesis isn't just one thing. It has two main stages. One that needs light directly.
The light-dependent reactions.
And another set of reactions that are controlled by enzymes and don't need light directly, though they depend on the light reactions products.
The light-independent or carbon-fixation reactions.
Yeah.
That's a key distinction we'll come back to. 2. The nature of light. Okay, history sorted. Now let's talk about the power source. Light. It all starts with Sir Isaac Newton, right? Showing white light isn't just white.
Exactly. Pass it through a prism and you get the rainbow, the visible spectrum of colors.
And then James Clerk Maxwell showed This visible spectrum is just a tiny fraction of a much bigger thing.
The electromagnetic spectrum. Radio waves, microwaves, X-rays. Light is just the part we see.
And it travels in waves, the key thing being wavelength.
Right, and wavelength is inversely related to energy. Shorter waves, like violet light, have more energy. Longer waves, like red light, have less.
Almost twice the energy in violet compared to red, I think the book said.
Something like that, yeah. But the wave idea didn't explain everything. There was this puzzle, the photoelectric effect.
Ah, right. Shine ultraviolet light on zinc and electrons pop off.
But here's the weird part. It didn't matter how bright the UV light was. Below a certain wavelength, nothing happened. It was the color or wavelength, not the intensity that mattered.
Which didn't fit the wave model.
Not at all. Then came Einstein. He proposed light also acts like particles, discrete packets of energy, photons or quanta, and the energy of a photon depends on its wavelength, just like the photoelectric effect showed. Shorter wavelength, higher energy photon.
So light is both.
Yeah.
A wave and a particle.
Yep. We need both models to fully get it. It's called wave-particle duality.
Now, why is visible light the key for life? The book calls it the fitness of light.
It's fascinating. Higher energy stuff like UV or X-rays is too powerful. It just breaks molecules apart, damages DNA. Dangerous.
And lower energy, like infrared.
Mostly just gets absorbed by water and generates heat. It doesn't have enough energy to sort of kickstart chemical reactions in the same way.
So visible light is in that Goldilocks zone.
Exactly. Just the right amount of energy to excite electrons in molecules to get things moving biologically, but without causing destruction.
And it's also the main type of light that actually reaches Earth's surface after filtering through the atmosphere.
Which is amazing when you think about it. Is that just luck? Or is life exquisitely tuned to the light available? It makes you wonder. 3. The role of pigments.
Okay, so we've got the right kind of energy, visible light. How do plants actually grab it?
Through pigments, molecules that absorb specific wavelengths of light.
And the pattern of absorption is called its absorption spectrum.
Right. And this explains why leaves are green. Chlorophyll, the main pigment.
It absorbs reds and blues really well.
But not green. It reflects green light, so that's the color we see.
Makes sense. Now, how do we know chlorophyll is actually doing the work in photosynthesis?
That's where the action spectrum comes in. You measure how effective different wavelengths are at driving the process, in this case, making oxygen.
Like that Engelmann experiment with the algae and bacteria.
Classic experiment. He's shown a spectrum of light onto a filament of Spirogyra algae. Then he added bacteria that love oxygen.
And they all clustered around the parts lit by red and blue light.
Exactly, because that's where the algae were photosynthesizing the most, releasing the most oxygen. The action spectrum matched chlorophyll's absorption spectrum. Proof positive.
So what happens physically when a pigment like chlorophyll absorbs a photon?
An electron within the molecule gets boosted to a higher energy level, an excited state.
And that energy doesn't just stay there, right?
No, it's unstable.
Yeah.
It has a few possible fates. One, it can just fall back down, releasing the energy as heat and maybe a less energetic photon that's fluorescence.
You can actually see chlorophyll fluoresce red sometimes.
You can. But for photosynthesis, that's wasted energy. The useful fates are 2. Resonance energy transfer. The energy, but not the electron itself, gets passed to a neighboring pigment molecule, like a tiny energy bucket brigade.
Okay, and the third way?
Electron transfer. The excited, high-energy electron itself physically moves to another molecule, an electron acceptor. This oxidizes the chlorophyll, leaving it short an electron.
And those last 2, resonance transfer and electron transfer, are the ones that actually drive photosynthesis forward.
Precisely. They channel the captured light energy into the chemical reactions.
And where does all this happen? Inside the chloroplasts, right? In those membrane systems.
Yep, the thylakoid membranes. Chlorophyll molecules aren't just floating around. They're precisely arranged with proteins within these membranes.
Sometimes stacked up into grana.
Right, grana stacks within the stroma fluid. And remember, the least structure helps too. Stomata let CO2 in, vascular bundles bring water and take sugars away.
So who are the main players, pigment-wise? Obviously chlorophyll.
Chlorophyll A is the absolute star. It's essential in all oxygen-producing photosynthesis. It's the one in the reaction center that actually loses the electron.
But there's also chlorophyll B.
Yes, and chlorophyll C in some algae. These are accessory pigments. They absorb light at slightly different wavelengths than chlorophyll A and pass the energy along to it via resonance transfer. Broadens the usable light spectrum.
Okay, and then there are the carotenoids.
The reds, oranges, yellows, also accessory pigments passing energy to chlorophyll, but they have another really critical job. They act as antioxidants. They protect chlorophyll from getting damaged by too much light energy, preventing oxidative stress.
Ah, so that's why we see them in the autumn. The chlorophyll breaks down, and the carotenoids that were there all along become visible.
Exactly. And finally, there are phycobilins, water-soluble pigments found in cyanobacteria and red algae, also helping capture light. 4. The reactions of photosynthesis.
All right, we have light, we have pigments capturing it in the chloroplasts. Now for the main event, the reactions themselves.
2 major stages, right? Yep. First, the light reactions, or energy transduction reactions. They need light directly. They capture light energy, use it to make ATP.
The energy currency?
And NADPH.
To reducing power,
Yeah.
carrying high-energy electrons.
And importantly, they split water and release oxygen. That's stage 1.
These two.
The carbon-fixation reactions, sometimes called the light-independent reactions, though they rely on the products of the light reactions.
So they use the ATP and NADPH?
To take CO2 from the air and convert it into sugars, building the actual organic molecules.
Yeah.
2 photosystems are involved in the light reactions.
Okay, let's dive into those light reactions. They happen in units called photosystems.
Right. Think of them as organized teams of pigments and proteins in the thylakoid membrane. Each one has like 250 to 400 pigment molecules.
And 2 main parts.
An antenna complex and a reaction center. The antenna complex is like a net full of chlorophyll A, B, and carotenoids gathering photons.
And they pass the energy inwards.
Through resonance transfer, yeah, funneling it down to the reaction center.
Which has a special pair.
A special pair of chlorophyll molecules. They're unique. In Photosystem I, it's called P700 because it best absorbs light at 700 nanometers. In Photosystem II, it's P680.
And this is where the electron transfer actually happens.
Yes. When energy reaches this special pair, one of its electrons gets so energized it jumps off onto a primary electron acceptor molecule. That kicks off the electron flow.
So we have Photosystem II, P680, and Photosystem I, P700. Why numbered that way?
Just the order they were discovered, not the order they work in. They usually work together, linked by an electron transport chain.
And they're not right next to each other in the membrane.
Interestingly, no. PS2 is mostly found in the granostacks, while PSI is mainly in the thylakoids connecting the grana, the stroma thylakoids.
So they need little messengers to carry electrons between them.
Exactly. Modal electron carriers are essential. B water is oxidized to oxygen by Photosystem II.
Okay, let's trace the path, starting in Photosystem II. P680 gets hit by light energy.
An electron gets excited, jumps off to the primary acceptor, pheophytin actually, then gets passed along a short chain, including plastiquinone molecules.
And this is where the water comes in.
This is the incredible part. P680 has lost an electron. It needs a replacement. Photosystem II has a unique component called the oxygen evolving complex.
Made of proteins and manganese ions.
Yep. And its job is to rip electrons out of water molecules. It performs photolysis. 2 water molecules are split into 4 electrons, 4 protons, H+, and 1 molecule of O2 gas.
Wow. So the electrons replace those lost by P680.
Keeping the process going. And the oxygen is released. That's the oxygen we breathe. And crucially, those protons are released inside the thylakoid space, the lumen.
Starting to build up a concentration gradient.
Exactly. We'll see why that's important very soon. C. The cytochrome B6F complex links photosystems to second I.
Okay, so the electron carrier plastiquinone, which picked up electrons from PS2 and protons from the stroma, where does it go?
It shuttles those electrons over to the next major component, the cytochrome B6F complex.
This sounds familiar. Like mitochondria.
Very analogous to complex 3 in mitochondria, yes. As electrons pass through it, this complex pumps more protons from the stroma into the thylakoid lumen.
Adding to that gradient even more.
Right. Then, the electrons are passed from the cytochrome complex to another mobile carrier. Plastocyanin, a small copper-containing protein, and its job is to ferry the electrons over to photosystem ID. ATP is synthesized by an ATP synthase complex.
Okay, before we get to PSI, let's talk about those protons. We've pumped a bunch into the thylakoid lumen from water splitting and the cytochrome complex.
So you've got this high concentration of protons inside the lumen and a lower concentration outside in the stroma. It's an electrochemical gradient, potential energy stored up.
Like water behind a dam.
Perfect analogy. And just like a dam has turbines, the thylakoid membrane has ATP synthase complexes.
These are enzymes.
Huge enzyme complexes that act like channels. They allow the protons to flow back out of the lumen down their concentration gradient into the stroma.
And that flow powers ATP production.
Yes. The energy released by the proton flow drives the synthesis of ATP from ADP and phosphate. It's called chemiosmosis, or chemiosmotic coupling.
Same fundamental process used in mitochondria and even bacteria.
Amazing. It really is a universal energy coupling mechanism. NADP plus is reduced to NADPH in Photosystem I.
All right, back to the electrons. Plastocyanin deliver them to Photosystem I, right, to replace electrons P700 lost.
Exactly. So now light hits P700 and PSI. An electron gets excited, jumps off, and goes down another shorter electron transport chain.
Involving carriers like ferredoxin.
Yes, ferredoxin is a key player here. It's an iron-sulfur protein. It accepts the high-energy electron from the PSI chain.
And what does it do with it?
Ferredoxin then transfers the electron to our final electron acceptor in this chain, NADP plus C.
Which needs electrons and a proton to become NADPH.
Right. An enzyme called ferredoxin NADP plus reductase, FNR, catalyzes this step. So NADP plus electrons plus a proton from the stroma equals NADPH.
Okay, so let's recap this whole flow. Electrons start at water, go through PS2, the cytochrome complex, PSI, and end up on NADPH.
That's the standard pathway, yes. It's called non-cyclic electron flow because the electrons move in one direction, from water to NADPH.
And along the way, we've produced oxygen, generated that proton gradient to make ATP
via non-cyclic photophosphorylation.
And produced NADPH.
Yeah.
All powered by light.
F-cyclic photophosphorylation generates only ATP.
But you mentioned non-cyclic. Does that mean there's a cyclic way too?
There is. Sometimes Photosystem I can work on its own in a process called cyclic electron flow.
How does that work?
Instead of passing electrons from ferredoxin to NADP plus egg, they get shunted back to the cytochrome B6F complex.
The one that links PS2 and PSI.
The very same. So the electrons flow from the cytochrome complex back through plastocyanin to P700 and PSI, get re-energized by light, and go around again. A cycle.
Okay, so electrons are just cycling through PSI and the cytochrome complex. What's the point?
Well, as electrons pass through the cytochrome complex, it still pumps protons into the lumen.
Ah, so you still build the proton gradient.
Exactly. And that proton gradient still drives ATP synthesis via ATP synthase. So cyclic electron flow makes ATP. We call this cyclic photophosphorylation.
But no NADPH is made, and no water is...
Correct. No NADPH production and no oxygen release. It only generates ATP.
Why would the plant need to do that? Just make extra ATP.
Precisely. As we'll see when we get to carbon fixation, the Calvin cycle actually uses more ATP than NADPH, roughly a 3.2 ratio. Non-cyclic flow produces them in roughly equal amounts.
So cyclic flow provides the extra ATP needed to balance the books for sugar production.
That's the main idea. It fine-tunes the ATP and ADPH ratio. It also raises an interesting evolutionary question. Maybe the earliest forms of photosynthesis only had something like PSI and used cyclic flow just to make ATP.
Plausible. V, the carbon fixation reactions. Okay, light reaction's done. We've got our ATP and NADPH. Now for stage 2, carbon fixation. Using that energy to build sugars from CO2.
Right. This happens out in the stroma, the fluid-filled space inside the chloroplast. And the CO2 gets into the leaf through those tiny pores, the stomata. In the Calvin cycle, CO2 is fixed via a 3-carbon pathway.
The main process here is the Calvin cycle, named after Melvin Calvin, naturally.
Nobel laureate for figuring it out. It's a cycle, meaning it starts and ends with the same molecule regenerating it along the way.
And that starting molecule is?
Ribulose 1,4,5-bisphosphate, or RuBP, a 5-carbon sugar with 2 phosphate groups attached.
Okay, so the cycle has 3 main parts.
Yep. First, fixation. This is where CO2 actually enters the cycle. The enzyme RuBP carboxylase oxygenase, better known as Rubisco.
The most abundant enzyme on Earth, apparently.
It is. Rubisco attaches 1 molecule of CO2 to the 5-carbon RuBP. This makes a very unstable 6-carbon intermediate,
which immediately breaks down.
Instantly splits in half to form 2 molecules of a 3-carbon compound called 3-phosphoglycerate, or PGA. This is the first stable product, hence why the Calvin cycle is also called the C3 pathway.
Okay, fixation done. Step 2.
Reduction. The PGA molecules are converted into another 3-carbon compound, glyceraldehyde 3-phosphate, or PGAL. This step uses up the ATP and NADPH generated during the light reactions. Energy input here.
Got it. And the third step?
Regeneration. This is crucial. For every 6 molecules of PGAL produced, only 1 represents net gain that 1 leaves the cycle to become sugar. The other 5 PGAL molecules are rearranged and use more ATP to regenerate the 3 molecules of RuBP we started with.
So the cycle can continue. It takes 3 turns of the cycle to fix 3 CO2 and produce 1 net PGAL.
Exactly. And the cost for that 1 net PGAL is significant. 9 ATP and 6 NADPH.
Ah, there's that 3.2 ratio of ATP to NADPH needed. That's why cyclic photophosphorylation is important for that extra ATP.
Makes perfect sense now, doesn't it?
So what happens to that PGL that leaves the cycle?
It's versatile. It can be exported out of the chloroplast into the cytoplasm and used to make sucrose table sugar, basically, which is the main sugar transported around the plant. Or it can stay in the chloroplast and be converted into starch for temporary storage. B. Photorespiration occurs when Rubisco binds O2 instead of CO2.
Now, you called the enzyme Rubisco, Ruby-P carboxylase oxygenase. Does the oxygenase part matter?
Oh, it definitely matters. It's a major complication. Rubisco isn't perfectly specific for CO2. Oxygen gas, O2, can also bind to its active site.
It competes with CO2.
It does. And when Rubisco binds O2 to Ruby-P instead of CO2, it initiates a process called photorespiration.
That's bad.
From a pure efficiency standpoint, yes. Instead of making 2 PGAs, it makes 1 PGA and 1 molecule of phosphoglycolate. This phosphoglycolate then enters a salvage pathway that actually consumes oxygen, releases previously fixed CO2, and uses ATP. It undoes some of the work of photosynthesis.
So it wastes energy and loses carbon. Why does it even happen?
It's favored when the concentration of O2 inside the leaf is high relative to CO2.
Like on hot, dry days when the plant closes its stomata to save water.
Exactly. Stomata close, CO2 levels drop inside the leaf, but photosynthesis keeps producing O2 from the light reactions, so the O2-CO2 ratio climbs. Prime conditions for photorespiration.
It can waste up to 50% of fixed carbon in some C3 plants? That seems incredibly inefficient.
It does. Though, to be fair, the salvage pathway does recover about 75% of the carbon from that phosphoglycolate, which is toxic if it builds up. And some argue photorespiration helps protect the plant from light damage under stress. But still.
Still seems like a flaw. Did Rubisco evolve when Earth's atmosphere had less oxygen, maybe?
That's a leading hypothesis. When Rubisco first evolved, atmospheric O2 was much lower, so its lack of specificity wasn't such a big deal. Now it is. See? The 4-carbon pathway is a solution to photorespiration.
So if photorespiration is a problem, especially in hot climates, did evolution find a workaround?
It certainly did. In some groups of plants, we see the C4 pathway. Think corn, sugarcane, many tropical grasses.
How does C4 work?
It's basically a CO2 pump. It uses an extra set of steps to concentrate CO2 around Rubisco, minimizing the oxygenase reaction. It involves a clever spatial separation.
Okay, break it down.
Step 1 happens in the outer leaf cells, the mesophyll cells. CO2 first combines with a 3-carbon molecule called phosphenol pyruvate, or PEP. The enzyme here is PEP carboxylase.
Rubisco.
No, PEP carboxylase. And crucially, PEP carboxylase has a very high affinity for bicarbonate, which CO2 becomes in water and is not bothered by oxygen at all.
Smart move. So CO2 plus PPP makes?
Oxyl acetate, a 4-carbon compound, hence the name C4 pathway.
Then what?
The oxaloacetate is usually converted to another 4-carbon molecule, malate. This malate then travels from the mesophyll cell into specialized cells deeper in the leaf, surrounding the vascular tissues, the bundle-sheath cells.
Okay, malate moves into the bundle-sheath cell.
And there, it's decarboxylated. The CO2 is released. Now you have a high concentration of CO2 right where the bundle-sheath cells keep their Rubisco and run the normal Calvin cycle.
Ah, so they pump CO2 into the bundle-sheath cells to feed Rubisco, keeping the O2 level relatively low there.
Exactly. It effectively eliminates photorespiration. The pyruvate left over from releasing the CO2 goes back to the mesophyll cell to be regenerated into PEP, which costs some ATP.
This structure mesophyll cells outside, bundle-sheath cells inside. Is that the Kranz anatomy?
That's the typical Kranz or wreath-like anatomy you see in most C4 leaves, though some plants manage C4 within a single cell using different compartments.
So what's the benefit? It costs extra ATP to run the pump?
True, it's energetically more expensive overall, but in hot, sunny, dry conditions where photorespiration would cripple a C3 plant, C4 plants thrive. Their net photosynthesis rates can be 2 to 3 times higher. They also use water and nitrogen more efficiently.
Like crabgrass taking over a lawn in summer.
Perfect example. The C3 lawn grass struggles with photorespiration in the heat. The C4 crabgrass doesn't care. D plants, having crassylation acid metabolism, can fix CO2 in the dark.
Okay, C4 uses spatial separation. Is there another way to beat photorespiration and save water?
There is. Found often in succulents, cacti, pineapples. It's called crassylation acid metabolism, or CAM.
CAM plants, what's their trick?
Temporal separation. They separate the steps in time, not space.
How does that work?
At night, when it's cooler and more humid, CAM plants open their stomata. They take in CO2 and fix it using PP carboxylase, just like C4 plants, forming oxaloacetate, then malate.
Okay, but then what?
They store this malate, as malic acid, in the large central vacuoles of their cells. All night long, they accumulate this stored acid. Makes the plant taste quite sour if you were to bite it early morning.
Got it. Storing CO2 as acid overnight. Then what happens during the day?
During the hot, dry day, they close their stomata completely to conserve water. Then they break down that stored malic acid inside their cells, releasing the CO2.
And that released CO2 feeds into?
The Kelvin cycle. Rubisco takes over, using ATP and NADPH generated by the light reactions happening during the day, all safely behind closed stomata. The acidity drops as the day goes on, so they taste sweeter later.
Wow. So they collect CO2 at night, use it during the day. That must save a lot of water.
Incredible amounts. They have the highest water use efficiency of all plants. They can survive in extreme deserts where nothing else can. The trade-off is usually very slow growth. Each of the carbon fixation mechanisms has its advantages and disadvantages.
So C3, C4, CM. It sounds like there's no single best way.
Absolutely not. Each strategy has costs and benefits. C3 is most efficient in cool, moist conditions with normal CO2. C4 excels in heat, highlight, and dryness, but costs more energy and can be cold sensitive. CM is the ultimate water saver, but typically grows very slowly.
It really shows how plants have adapted in amazing ways to different environments. F, global warming. The future is now, connecting to the bigger picture. And this whole discussion about CO2 levels and plant responses obviously connects directly to global warming, doesn't it?
It's highly relevant. The scientific consensus is clear. Earth temperature is rising, driven by human activity releasing greenhouse gases, especially CO2.
How might rising CO2 affect these different plants?
Well, you might think C3 plants would benefit most, right? Higher CO2 could reduce their photorespiration problem, potentially boosting growth, the so-called CO2 fertilization effect.
Would that give them an edge over C4 plants?
Potentially. C4 plants might lose some of their relative advantage if CO2 levels keep climbing, as their CO2 concentrating mechanism becomes less critical. But its complex temperature, water availability, nutrients all interact. The effects on agriculture and natural ecosystems could be huge, not to mention impacts like rising sea levels.
Which raises the critical question. What do we do?
The consensus goal is to reduce atmospheric CO2. That means tackling fossil fuel use, developing alternative energy, and crucially, protecting and expanding forests, which are massive carbon sinks. Understanding plant physiology is actually vital for tackling climate change. Outro.
Okay, that was quite a journey. We went from ancient Greeks thinking plants ate soil,
There's centuries of experiments uncovering gas exchange, the role of light, the source of oxygen.
to the intricate dance of photosystems, electron transport chains, ATP synthesis.
And finally, the different ways plants fix carbon, C3, C4, CM each, a brilliant adaptation.
It really underscores how this one process, photosynthesis, underpins almost everything we see in the living world.
It does, and understanding it isn't just academic. As we face challenges like climate change, knowing how plants work, how they adapt, how they interact with the atmosphere, it's critical knowledge. Their adaptability is maybe the biggest lesson here.
So a final thought to leave you with. As our environment continues to change, how might plants adapt further? Could we see new photosynthetic strategies evolve? And how can our deeper understanding help us navigate that future?
Lots to think about.
Indeed. Thank you for joining us on this deep dive into the raven biology of plants. We hope it's given you a valuable shortcut to understanding this vital topic and maybe sparked some curiosity to learn even more.